Okay, Assalamu alaikum. Today we want to continue talking about carbohydrates. We reach into this slide, linear and branch chain polymers of alpha-D glucose. Notice that here we have the linear polyglucose chain occurs in amylose. All glycosidic bonds are of the type alpha-1,4. However, in the branch chain polymer occurs in amylopectin. Notice here that all the linkages are alpha-1,4 except for the branch here, and this is alpha-1,6. This occurs in amylopectin since starch consists of amylose and amylopectin. Now also here in the next slide we want to show you reducing sugars. This is a disaccharide with a free hemiacetal end. Is a reducing sugar because of the presence of a free anomeric aldehyde carbonyl or potential aldehyde group. Now this is called the reducing end, and this is called the non-reducing end. So notice that reducing sugars, they give positive tolerance test by formation of the silver mirror inside the reaction container. Now, amino sugars are an interesting class of compounds related to monosaccharides. And in the amino sugars, here we have two examples. They are important examples. We have at the beginning, if we substituted the OH group at carbon 2 by NH2, it's called glucosamine. And if we substitute the hydrogen or nitrogen with the acetyl group, it becomes N-acetyl beta-D-glucosamine. And the other one is we replace the OH with pyruvic acid. It's called N-acetyl muramic acid. These two compounds, as we will see, are components of bacterial cell walls. Now, summary of the reactions of monosaccharides. Sugars can undergo oxidation reaction and form esters. Glycosidic linkages are responsible for the bonding of monosaccharides to form oligosaccharides and polysaccharides. Now, some important oligosaccharides, as we will see. There are oligomers of sugars frequently occur as disaccharides. They form by linking two monosaccharide units by glycosidic bonds. And here we can see this figure shows us the structures of several important disaccharides. Notice that the notation HOH means that the configuration can be either alpha or beta. When a D sugar is drawn in this orientation, if the OH group is above the ring, the configuration is termed beta. The configuration is termed alpha if the OH group is below the ring, as we said before. Also note that sucrose has no free anomeric carbon atoms. So all these disaccharides we will talk about in lactose, maltose, sucrose, silobiose, and isomaltose. Notice that all of them has a free reducing end except for sucrose. So sucrose is not a reducing sugar. Okay. Now let's talk about each one. Sucrose, the common table sugar, which is obtained from the juice, and sugar beets, disaccharide form monoglucose and fructose at this time bonded together. Then the monosaccharides are alpha-D glucose and beta-D fructose, and the glycosidic linkage notation is alpha-beta-1,2. That's why it's not a reducing sugar, no free anomeric carbon. Both of them are involved in glycosidic linkage. In addition to sucrose, we have also fructose, and ketosis in general can act as reducing sugars because they can isomerize to aldoses. Now, we have sucrose is a table sugar and it is a common sweetener, but we have, remember, we have artificial sweeteners have been produced in the labs, etc. For example, saccharin cause cancer, as well as cyclamates. Aspartame has been suspected of causing neurological problems. Another artificial sweetener, which is derivative of sucrose, is called sucralose. Sucralose is, however, safe. General acceptance of the artificial sweetener aspartame, sucralose, and saccharin has led many dietitians to question the health benefits when their use does not appear to provide caloric saving, etc. So, we recommend here sucralose. Now, lactose, milk sugar made up of, as you can see, beta D-galactose and D-glucose. This is D-glucose and this is beta D-galactose. Remember that galactose is a carbon 4 isomer of glucose, as we mentioned in the last lecture. The type of linkage is beta 1,4. It is also a reducing sugar, as well as maltose, the disaccharide that is obtained from the hydrolysis of starch, also consists of two P-glucose joined together by alpha 1,4 linkage. However, maltose differs from cellulose in the type of linkage. This is beta 1,4 in cellulose and alpha 1,4 in maltose. Cellulose is obtained from the hydrolysis of cellulose. Okay. These are the two difference. The difference between, although they have the same monosaccharide, but they differ in the type of linkage, however. So in summary here, sucrose, a disaccharide, is common table in common sugar, consists of glucose and fructose linked by a glycosidic bond. Two other common disaccharides include lactose, which is found in milk, and maltose, which is obtained from starch. Also, we have isomaltose, and it is also consists of two glucoses, but the type of linkage is alpha 1,6. So it occurs at the branch point. Now we come to the so-called structures and functions of polysaccharides. Now, they are polymers of sugars. Glucose is the most common monomer, and they could be a polymer that consists only of one type of monosaccharide is called homopolysaccharide. A polymer that consists of more than one type of monosaccharide is termed as heteropolysaccharides. In this category, cellulose and chitin, which are structural materials, are polysaccharides with beta-glycosidic linkages. Starch and glycogen are polysaccharides with alpha-glycosidic linkage. They are carbohydrate storage polymers. Starch in plants and glycogen in animals. In the heteropolysaccharides, polymer consisting of more, as we will see, more than one type of monosaccharides. Let's start with cellulose, which is the major structural component of plants, especially of wood and plant fibers. It is, of course, a linear homopolysaccharide of beta D-glucose, and all the residues are linked by beta 1,4 linkages. Now, individual polysaccharide chains are hydrogen bonded together, giving plant fibers their mechanical strength. And here we have enzymes called cellulases. Animals lack the enzyme called cellulases that hydrolyze cellulose to glucose. So we cannot, human cannot digest cellulose. However, termites do, and the grazing animals, they harbor bacteria. They inhabit, and the bacteria that inhabit the digestive tracts of insects such as termites and the grazing animals such as cattle and horses. So they are able to digest cellulose, but man cannot. Even though animals cannot digest cellulose, this polymer still plays an important role as a component of dietary fibers. Stimulates the peristaltic action and thus help move the digested food through the intestine. Also, toxic substances in food and in the fluid bind to fiber and are exported from the body, thus preventing them from damaging the lower intestine and being reabsorbed there. Also, statistical evidence indicates that high fiber also reduces colon and other cancers, because fiber binds suspected carcinogens. Okay, fiber does bind also to cholesterol. Then again, fiber does bind to cholesterol and certainly causes some decrease in the amount of cholesterol in the blood. Fiber come in two forms, soluble and insoluble. The most common insoluble fiber is cellulose, which is found in lettuce, carrots, celery, brown rice, most other vegetables, many fruit skins, etc. Insoluble fiber binds various molecules, but otherwise merely forms bulk in the lower intestine. Soluble fibers include amylopectin and other pectins, as well as complex starches. Now, fibers seem to be more beneficial because of their increased surface area. Good sources include bran, barley, fresh fruits, potatoes, and skin beans, zucchini. However, soluble fiber often binds water very well. That's why they increase satiety by helping to fill the stomach. This is the importance of fiber. Now, if we look at this figure, which shows us the polymeric structure of cellulose, notice here beta cellulose is the repeating disaccharide. The monomer of cellulose is beta anomer of glucose, which gives rise to long chains that can hydrogen bond. You see, that can hydrogen bond to each other and give mechanical strength to cellulose. And as you can see, cellulose resembles beta sheet, whereas amylose with this helical structure resemble alpha helix in proteins. Then this structure also shows us the structure of a starch is based on the alpha anomer of glucose. The monomer is, as I said, alpha glucose, which gives rise to a chain that folds into a helical form. The repeating dimer has an alpha 1,4 linkage throughout. So remember, then cellulose resemble beta sheet and amylose resemble alpha helix. Now we come to starches. We talked about the two types. Polymers of alpha-D-glucose that occur in plant cells as starch granules in the cytosol. Types can be distinguished from one another by their degree of chain branching. Amylose linear, amylopectin branched. And I showed you before that, the alpha 1,4 and alpha 1,6. And here in this linear form, you see amylose with all bonds alpha 1,4. Now in amylopectin, in addition to alpha 1,4, as you can see, we have here at the branch alpha 1,6. So at the branch point, we have only alpha 1,6, as you can see. Now amylopectin is highly branched, with the branches occurring at every 12 to 30 residue. Now, there is something called starch-iodine complex. The most usual conformation of amylose is a helix with 6 residue per turn. يعني هي 1, 2, 3, 4, 5, 6. Iodine molecules, I think, can fit inside of the helix to form a starch-iodine complex, which has a characteristic blue color, dark blue. Then, most usual conformation of amylose is a helix with 6 residues per turn. Iodine molecules fit here, which has dark blue. Now, the formation of this complex is a well-known test for the presence of a starch. So, iodine test is to test the presence of starch. Now, also, amylopectin gives a positive iodine test, but the color is red rather than blue, so you can distinguish between them. Can distinguish between amylose and amylopectin. And the color is red-brown. Now, because starches are storage molecules, there must be a mechanism for releasing glucose from starch when the organism needs energy. Now, both we have 2 of these enzymes. Both the plants and animals contain enzymes that hydrolyze starch, and the alpha and beta amylase attack alpha 1,4 linkages. Here, beta amylase is an exoglycosidase that cleaves from the non-reducing end of the polymer, whereas maltose, the dimer of glucose, is the product of this reaction. And the alpha amylase is an endoglycosidase that hydrolyzes glycosidic linkages anywhere along the chain. Glucose and maltose are the products of the reaction. Now, amylose can be completely degraded by these 2 amylases, but amylopectin is not completely degraded because of the branch point. So, there are debranching enzymes which are also found in both plants and animals. They tend to degrade this alpha 1 linkages. So with these enzymes, when these 2 enzymes, the amylases and the debranching enzymes, are combined, they contribute to the complete degradation of both forms of starch. Now, how is the glycogen related to starch? Now, although starches occur only in plants, there is a similar carbohydrate storage form in animals called glycogen. Glycogen is a branched chain polymer of alpha-D-glucose, allowing a quicker mobilization of a glucose. It consists of a chain of alpha 1,4 linkages with alpha 1,6 linkages at the branch point. Now, the main difference between glycogen and amylopectin is that glycogen is more highly branched because the branch points occur every 10 residue in glycogen and every 25 residue in amylopectin. Let's look at the the structure of both amylopectin and glycogen. You see which is more highly branched? Glycogen. Why? Because branches occur at every 10 glucose residue, whereas in amylopectin branches occur at every 25 residue. Now, also you can distinguish between these 2 by the iodine test. Amylopectin gives a red-brown color, whereas the glycogen give a reddish color, very reddish color. When an organism needs energy quickly, the glycogen phosphorylase, this is an enzyme, has more potential targets if there are more branches, allowing a quicker mobilization of glucose. إذن لاحظوا, this glycogen, which is stored mainly in the muscle and to some extent in the liver, when there is a need for energy, there is an enzyme called glycogen phosphorylase that breaks down all these branches at one time, وبالتالي there is allowing a quicker mobilization of glucose. So this is the importance of the structure of glycogen. Now, in the heart of every glycogen molecule, there is an enzyme or protein called the glycogenin. Protein situated at the heart of every glycogen is found in animal cells in granules and are observed in liver and muscle cells. Now, glycogen phosphorylase and the debranching enzyme together assist the breakdown of glycogen, as we will see in chapter 18 when we reach there. Very good. Then after that, we go to the so-called linear homopolysaccharide of glucose derivatives. What is chitin? Chitin is a linear homopolysaccharide with all the residues linked at beta 1,4 glycosidic bonds, differs from cellulose in the nature of the monosaccharide unit. In cellulose, the monomer is beta-D-glucose. However, in chitin, the monomer is N-acetyl beta-D-glucosamine. It is a derivative of glucosamine. And this is the substitution of N-acetyl amino group for the hydroxyl group of carbon 2 in glucose. This is the substitution. Now, like cellulose, chitin plays a crucial role and has a fair amount of mechanical strength because the individual strands are held together by hydrogen bonds. Now, where do you find chitin? Major structural component of the exoskeletons of invertebrates, such as insects and crustaceans. However, it also occurs in cell walls of algae, fungi, and yeasts. Now, this figure shows you the polymeric structure of chitin. لاحظوا the monomer called N-acetyl glucosamine. This is the monomer, and a dimer of N-acetyl glucosamine is the repeating disaccharide. We call this the repeating disaccharide. Two molecules of N-acetyl beta-D-glucosamine are joined together by beta 1,4, and this dimer is repeated on and on and on. Now we come to the so-called heteropolysaccharides. Heteropolysaccharides are major components of bacterial cell walls. Now, prokaryotic cell walls contain polysaccharides cross-linked by peptides. Repeating unit of polysaccharide consists of two residues held together by beta 1,4 glycosidic linkage. You see? One of the two monomers is N-acetyl D-glucosamine. Here, you notice here substitution of a lactic acid side chain for the hydroxyl group, as I said, on carbon 3. And the other one is N-acetyl uramic acid. Yes, this is the structure of N-acetyl muramic acid, and this is the structure of N-acetyl glucosamine. You see, they are different from each other by this group. This is the difference. Substitution of lactic acid instead of OH at carbon 3. 1, 2, 3. لاحظوا here, this side chain here, this is called lactic acid. So this is the only difference between them, NAM and NAG, and they are abbreviated NAM and NAG, as you can see. Now the cross-links in bacterial cell wall consist of small peptides. Okay, as we are going to see in this figure, لاحظوا here, in this figure, in the cell wall of the bacterium Staphylococcus aureus, an oligomer of 4 amino acids, a tetramer, is bonded to N-acetylmuramic acid, forming side chain here. Then at the beginning here, this is the structure of the disaccharide, and here we have bonding of a tetrapeptide on the lactic acid here. Now the tetrapeptide forms 2 cross-links, both of them to a pentapeptide that consists of 5 glycine residue. Then again here we have, in addition to the tetrapeptide, لاحظوا 2 pentaglycines are bonded here. 2 pentaglycines. One is to the N-terminal group of D-alanine, and the other one to the epsilon amino group of lysine residue that is formed in the لاحظوا. This is the repeating disaccharide of the tetrapeptide side chain اللي هو in red, contains L-alanine, D-glutamine, L-lysine, D-alanine, and then we have C double bond or O minus. So after that, 2 pentaglycines are bonded, one to the epsilon amino group of lysine, and the other is bonded to the C-terminal end of alanine. And here you can see this is schematic diagram of the peptidoglycan. So peptidoglycan is the structure that is found in bacterial cell wall. Okay, then the material that results from the cross-linking of polysaccharide bipeptides is a peptidoglycan هذا. So named because it has both peptide and carbohydrate components. Notice here the cross-linking. Now the larger spheres هذول, they are the glucose units, and لاحظوا the red ones are the tetrapeptide, and the blue ones are the pentaglycine. And as a result of this all cross-link, this will give a mechanical strength to bacterial cell wall. So that's why bacterial cell walls are extremely difficult to disrupt. Now, then this is peptidoglycan again. Okay, we talked about that. The same. Notice here we have this blue N-acetylglucosamine residue, and here N-acetylmuramic acid, the one in yellow. Now the red one, amino acid residue of the tetrapeptide, and the blue glycine residues of the pentapeptide side chain. Now we come to the so-called pectin. Now pectin is extracted. لاحظوا, plant cell walls consist largely of cellulose, but the other important polysaccharide component found in plant cell wall is called pectin. Now, pectin is a polymer made up mostly of this compound called D-galacturonic acid. This is a derivative of galactose. Galactose at carbon 6 CH2OH. We oxidize this alcohol to get this carboxylic acid. So this is the repeating monomer of pectin. Pectin is extracted from plants because it has commercial importance in the food processing industry as a gelling agent in yogurt, fruit preserves, jams, and jellies. Now, the major non-polysaccharide component in plant cell wall, especially in wood plant, is called lignin. Lignin, however, is a polymer of coniferal alcohol. This is the structure of coniferal alcohol, you see. And lignin from Latin lignum means wood. This is found especially in wood plant. And it is very tough and durable material, but unlike bacterial cell walls, plant cell walls contain comparatively little peptide or protein. Little peptide or protein. So we will stop here for today, ladies and gentlemen, and if you have a question, please ask. Any question? Ask about anything. Remember your exam next Tuesday. 4 chapters are included as there is an ad in the e-learning. Chapters 3, 4, 6, and 7. Each chapter 10 multiple choice questions. And all the details about this, you can read it in the e-learning.
يعطيك العافية دكتور.
عافاك الله. Any question before the time ends? إذا next time يعني إن شاء الله we continue what left from this chapter a little, 2-3 slides, 4 slides, and then we go to the next chapter.